Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Wett, and I'm the founder of Everything Epigenetics. Today, my guest is Raghav Sehgal, and our main discussion will be his research looking at more of a systems approach to epigenetic aging. This is really cutting edge research. It is not yet published, so this is one you will definitely want to sit down and tune into. And Raghav is a PhD student at Yale University, presently solving this large aging issue. His research really focuses on understanding human aging and how we're aging at a molecular level. Through his research, he's trying to answer critical questions that we'll also be answering during the podcast today, such as what are the biological systems and processes driving human aging? How can we measure and capture this biological aging? Can we actually reverse the processes driving aging? We're also going to talk about how aging can give rise to different diseases, such as cancer, diabetes, Alzheimer's, and more. And during his time at Yale, to answer these questions, Raghav is building deep learning tools and techniques using a multi-omic approach. This is where he's taking data from all of these omic realms, such as epigenetics, transcriptomics, proteomics, metabolomics, and more. And he's also using a multimodal data approach, where he's looking at whole body and organ biomarkers, tissue omics, and single cell omics too. He has 7 years of experience embedding data science and artificial intelligence to the biomedical space as a scientist, engineer, product manager, team builder, academician, and entrepreneur. And he was previously involved in building and managing a Series A funded biomedical data science startup, which we'll also chat about. Raghav is a Gruber Grant Awardee and an Impetus Aging Grant Awardee too. Now for my guest, Raghav Sehgal. Welcome to the Everything Epigenetics podcast, Raghav. I am so excited to have you today. We have a lot of exciting things to chat about.
Oh, thank you for having me here, Hannah. This is really exciting. And I'm very excited to chat with you today and I guess answer all your questions.
Yeah, I was just so curious about your work because a lot of it isn't available to the public. I'm very new myself to all of your research and super keen to learn more. I spoke with several people after they attended the Gordon Research Conference, as I mentioned to you when we were just chatting, and they talked very highly about the work that you're doing. And before we dive headfirst and jump in, I'd love to start by just getting to know you a little bit more. So tell me a little bit about where you are today, how you got there, your interests, and where that really peaked.
Yeah. So I've had a rather interesting journey and sort of tumbled my way into sort of the aging field, in all honesty. I did my engineering back in India. It was in electronics and communication engineering, a very different field. And during my undergrad, actually, I came across this really cool company called Elucidata. They were just kind of starting out and they needed someone who was like a data scientist. They were a computational biology company, sort of agglomerating publicly available data sets and selling them to pharma companies. And that's kind of where I sort of started getting involved in computational biology, vaguely speaking. And so I spent a couple of years of my life sort of working with them. The startup now is a fairly big startup. It's a Series A funded $100 million startup. So I'm fairly proud of it. And during that period, I also realized, though, that I'm mathematically very strong in sort of doing something like computational biology, given my engineering background. But when it comes to the actual biology of things, that's not really my strong point. And which is why I thought that it might be a great opportunity for me to sort of take this up in grad school and sort of invest more time into it. And so that's what I did. I joined Yale as a computational biologist. And initially, my plan was, I'll be working with like big omic data sets and finding new drugs and new drug targets. But on a very interesting seminar, which I wasn't planning to go to at all. A friend of mine dragged me to it, and just like any grad student, since there was free food, I agreed. Uh, uh, this seminar was actually being led by Morgan Levine, who was back then a PI, a professor at Yale, and yeah, she just kind of changed my perspective on aging. I never thought aging or as like a disease, so to speak of. I always thought it's kind of this natural process that happens to everyone. And I think that perspective change really drew me into the field and kind of ended up with me kind of rotating with Morgan and eventually joining her lab and sort of working on building all of these really cool epigenetic clocks that I'm now working on.
Yeah, that's awesome that you saw her speak and that was like your turning point in the career. And then you joined her lab and it opened you up into this entire new world, because I remember that turning point for me as well, where I had no idea this existed. And when people ask me about that, I can like remember when I was opened to this world of aging and preventative integrative functional medicine. So it's like weird to think of my life before when I didn't know about it. It's like all these possibilities.
It's really transformative in that sense, right? Like I always feel like some of these, in science, you always kind of feel like, hey, I'm doing this really cool thing, and this is kind of the best thing that the world can see, and then you experience this new kind of science, and you're like, oh my god, I didn't know this at all, and I can totally relate with that experience of yours, where you're like, mind blown away.
Yeah, such an epiphany. Well, no, I love hearing about your backstory. Always curious to see how people really get started in this field. So what we're going to talk about today is more of your systems aging clock. And I want to preface by saying a lot of our listeners are probably very familiar with, a lot of my listeners are probably very familiar with these epigenetic clocks, these biological aging clocks. And those are based on large scale meta-analysis. They come up with this biological age, this single metric. Sometimes, there's the pace of aging, that Dunedin clock. But, your work is influenced by this idea that the single biological age number is just not enough. And it may not provide enough feedback to what's happening on a biological level. So, you believe that this has to do a lot with the heterogeneity of aging. And I want to really get into that, have you talk about that, maybe define heterogeneity to begin, and then we'll go from there.
Yeah, no, that makes sense. So I'll kind of start with kind of the clocks that are out there. I think they're amazing.
Mhm.
Grimmage, Dunedin pace, Age, also like the first generation of clocks that were kind of predicting age and not sort of something like mortality or pace of aging. I think they're all amazing clocks, and they have provided us with such meaningful insights into how we're aging as individuals and sort of their relationships with different sort of these age accelerated relationships with different comorbidities has been quite impressive. And prior to sort of these clocks, there was never anything that could quite predict aging like these clocks can. So to start with, I stand on the shoulders of stalwarts in a way, right? These clocks are amazing, and they are so powerful. That being said, there's always space for improvement in anything that's out there, and I think that's kind of where we really started thinking about this. So these clocks, if conceptually speaking, what do? They take sort of whole blood methylation, right? And they use it as a proxy of your whole body aging, which is quite impressive. These numbers that we get are quite impressive. But here's the thing, as we know from biology in general, our organs and our different biological systems don't age at the same rate. If all of our biological systems were aging at the same rate, all of us will get the same diseases at the same time. So all of us will get cardiovascular disease, say at 60, and then all of us will get Alzheimer's at 80. But that's not the case. Our genetics, our sort of environmental factors influence the kind of aging that occurs in our body. And that kind of leads to kind of this differential aging across our different organs and biological systems. And this is the kind of sort of heterogeneity, which is what I'm talking about. So at one level, there's the heterogeneity, which is, across different individuals, aging rates are different. But within an individual itself, the organs and the biological systems age at very different rates. And so what we really wanted to capture was how are people or how are the different organs within an individual aging differently. And the reason we find that this is really valuable is because now, using this information, we believe that we can actually more accurately predict the kind of comorbidities, the kind of health span, the kind of lifespan a person would have. So, hypothetically speaking, uh, think about two individuals who have the same overall biological age, say calculated by Dunedin pace or Grimm age, but we'd really like to understand why do they have the same biological age, right? Is it because both of their hearts are really aged? Maybe one of their hearts is really aged, for someone else, their liver is really aged. And then the kind of interventions that you practice in this case would be very different. Say, if I have a really strong heart aging or really accelerated heart aging, I might want to take metformin, right? Hypothetically speaking, assuming if metformin does decrease cardiovascular age, and or if my brain aging is really high, I might want to take nicotinamide, which is supposedly another one which can slow brain aging. But I'm not saying you should take metformin or nicotinamide for any of those reasons.
Right.
But at the same time, I am saying that you can then practice more targeted interventions that can potentially lead down to slowing in specific organ systems, and that hopefully will be a lot more meaningful in actually being able to sort of decrease your overall biological age.
Yeah, very interesting. So was that a new thought that all of these organ systems are aging differently, or was that something that's been around for a while and that we were maybe a little bit aware of, but never have done much research around that?
I think we always kind of knew at some level that our different organ systems will age differently. There's canonical evidence in the fact that, as I said, as I was giving the example, not all of us die of the same disease at the same time, right? So clearly our organ systems are aging in different manners. And so there's always been kind of this canonical evidence, but for once, I think we have been able to quantitatively show that that's the case, that individuals do age differently in their different organs, and those different rates of aging are actually sort of quantifiable in a way, and from a very simple method, relatively speaking.
Yeah, I've measured my, methylation markers on the Epic 850K array several times. I'm waiting for my most recent data to get back, but now I want to know the age of my organs. Like that's the next best thing because sure, if your biological age is older, then you would definitely want to know what organs are maybe causing that acceleration. But if your biological age is younger, my assumption is you could still have some organs that are maybe aging a little bit older as well. So you would want to identify those organs that may be having a little bit of a quicker aging and take, set interventions or preventative approach to then, further decrease that biological age.
Exactly, right. And I think uh, what's really valuable out of this is that, uh, and probably we can talk a little more about this as we move forward, but beyond just the fact that different organs age differently, I think there's also like a combinatorial effect. So think about it like when a car starts to break down, uh, if your axle goes off, or if your pedal goes off, right, or if your brake goes off, or starts bearing off, that's not the only thing that will wear off. It will have like a domino effect of sorts, right?
Right.
And so you will not only just see that, hey, your heart aging is going really fast, but maybe your kidney aging, which is very closely tied into heart aging, is also accelerating at the same time. And that actually gets into a really interesting sort of caveat or of aging that we're now starting to call aging subtypes or agiotypes almost, right? That individuals, if you actually start looking at these sort of different measures that we have created and look at them together instead of independently, you can actually start seeing individuals being aging in almost very different patterns, having very different predisposition to diseases. And that's where it really gets interesting, because that is when you can really start intervening and saying that, hey, this is your aging subtype. Let's provide you with these medications so that we can slow down your aging subtype. And that to me is like really valuable.
Gotcha. So, you may have these aging organs, which is going to affect synergistically all together these other organs, right? Think of it all out on like a string, and they're pulling each other back and forth. And then based on that, you're classified or put into some type of agiotype, which could eventually have a treatment plan. And it all goes back to that preventative type of medical approach. No,
exactly.
I really like that. And let's get more on the computational side of what you did with your work, since that's really your bread and butter. So we have these different organ systems, or these functional domains that you call it, and you're using this system-based approach that we've been chatting about. So can you explain like how you worked through the body? Like what organ systems did you pick? You know, what made you choose those specific biological processes?
Yeah. So the from the get-go, what we did was we started with a more unsupervised approach where we said that, hey, what we're going to do is we're going to take a lot of these functional biomarkers and clinical chemistry biomarkers and cell sorting information from different individuals, and we are all going to throw it into like a dimensionality reduction technique and see what dimensions show up. And what that kind of gave us an insight into that these different biomarkers were kind of organizing themselves or sort of having similar dimensions to each other, depending on their on the sort of organs that they were related to or the biological process they were related to. So in practice, this was kind of almost like it was kind of like its own discovery that they were organizing themselves into these sort of behavioral patterns that were marked by these organs. And so that gave us the insight that, hey, that there are these different systems that we can really sort of are behaving very distinctly in a way, and we can really capture that information separately, at least based on very well-known biomarkers. And then the question was that, hey, if these systems are behaving very differently and have these sort of very distinct sort of trajectories, so to speak of, then is this information then being captured in our methylation data from our blood? And lo and behold, we built these kind of elastic net models to sort of predict these sort of trajectories of these organs, and they do actually segregate out. They're very different, as they are not sort of highly correlated and behave very differently. And are actually then we sort of even validate to show that they are very specific to the organ that they are built for. So it was partly, it was like, initially it was just like we tried things out and we saw that there were these 11 organ systems, biological systems that were showing up. And that being said, I also do sort of recognize that this is not the end all be all, that there are only 11 systems. It's partly a limitation of the data that we have as well, right? We have only so many biomarkers, even though we use like, I don't know, 160 biomarkers or something, it's still 160 biomarkers only. If we had hypothetically even more biomarkers or even more data, we might probably discover even more biological systems that were segregating out. But given the data that we have, we were able to find sort of these 11 distinct systems, and which we then trained using our methylation data to figure out these aging trajectories.
Sure. Yeah, I imagine it's very hard when you're looking at something so complex such as the human body and trying to break it down into these different systems or classify it, right? Like whenever anyone asks, hey, what do you do? It's just so hard to talk about like biological aging and DNA methylation and like where to start. So I couldn't imagine like trying to break down the body in these different ways. And just to name a few, correct me if I'm wrong, I think you have some like the immune function, metabolic function, hepatic function, cardiac, renal, and obviously, you know, a couple more. You said 11 and 160 biomarkers that fall into each of those categories. Were there any systems you would have liked to include that didn't make the list that you were just like, oh, darn? And I know you said, you know, you're working with the data that you have there, of course, which is what all of us need to do when creating a new project or looking at a new area of interest.
Yeah, no, I think in that sense, systems age is fairly sort of very well encompassing. That being said, there's always, as I said, scope for improvement in science. So as a limitation, I can tell you is that even though we have, for example, like a hormone system, We don't necessarily have a system that is able to capture sort of reproductive aging, which I believe is a very strong predictor of aging, especially in women and sort of in people of the female sex. And so I think it's one other sort of system that I would really like to capture in the future, because that would additionally sort of improve our understanding of the biology of aging in general. Another system that I feel that we could potentially additionally capture is kind of, again, the equivalent of that is kind of the male reproductive system. But another one which I feel is very important that could be additionally captured. So we have like a metabolic system and we have like the liver, but we don't have something which is very specific to the gut. And I know that recently, if you have seen the hallmarks of aging paper, dysbiosis has become like one of this new hallmark of aging. Sort of the gut microbiome has become this new fulcrum point of aging. And so that is one other sort of potential system that I would really like to capture at some point using this data. But again, there could be so many more that are out there. And as we have more data, we discover more of them. But yeah, I think just from the get-go, these are a few that I really think would be exceptionally well added to this list of system scores that we have.
Sure. Yeah, I know, fertility obviously is a huge studied kind of subgroup or category within DNA methylation. And then it was interesting and funny that you brought up the microbiome in the gut, because I get asked that question a lot from my healthcare providers. Like, do you know the relationship and methylation? And there's obviously definitely something there, but it's not really well studied with methylation. So I think a lot that we can uncover and learn. So very cool. You have these distinct groups of biological age processes. So you're taking these 11 systems and you have these biological scores, right, for each of these systems. So you kind of have, do you have these biological age clock then for those 11 systems, correct?
Yeah, that is correct. Yeah.
Perfect. And then you're taking those and you're combining them to create the system's aging clock, right?
Yeah, that is correct. Yeah.
Perfect. And what did you find there? Yeah, explain that process.
Yeah, no, I think so. So partly, I'll be very honest. I think the reason why we completely made sort of this overall aging clock, I think, is really useful because we would like to sort of compare it to the presently available clocks that are out there and show kind of whether we are able to perform as good or not, and so on and so forth. But I think the honest answer is that it is these individual scores that are really, or these individual sort of organ clocks that are the really meat of this whole exercise that we have done, the scientific approach that we have taken. And so the overall clock is really helpful in sort of telling you, hey, this is your overall age, this is what you look like. But then it is these kind of independent scores that really inform you that, hey, this is why your overall aging score is so high. So if you get, say, you're 2 years older than you are or supposed to be, the reason you're 2 years older is because your heart is 4 years older than it's supposed to be, but your liver is 2 years younger. So maybe don't focus on your liver, focus more on your heart aging, and so on and so forth. And so that's kind of really where it gets super interesting for me. I think the other thing that we really found, and I think that's very valuable, is so a part of our work was also sort of looking at a lot of the presently available clocks that are out there, and they're really good, like PhenoAge, GrimAge, Dunedin and Pace. They can capture sort of the way they are built. They capture sort of these different aging dimensions really well. But that's kind of the thing. They're able to capture maybe one or a few aging dimensions really well. But what we wanted to attempt was build a clock that is able to capture all of these different aging dimensions really well and kind of have a more balanced approach of sorts. Which is not to say that any of those clocks are limited in any way. I think they're all exceptional clocks. GrimAge predicts for cardiovascular disease and lung cancer really well. Dunedin Pace predicts for sort of musculoskeletal related issues, sort of frailty really well. PhenoAge predicts for a bunch of other things really well. But what you really want is kind of like a balanced score across them all. You don't want to be over predicting for one particular dimension. What you really want to be saying that, hey, my clock is not really over predicting for cardiovascular aging dimension excessively, but it's also predicting well for kidney dimension of aging or some other dimension of aging. And that's what we're in some of our sort of analysis, that's what we have really seen, that what systems age, because it's kind of using all of this systems approach of like integrating all of these different informations, we're really able to capture all of these different dimensions of aging in a more balanced fashion without being kind of biased in one particular direction.
And I think I agree with you. All of the clocks that have been created to date, the epigenetic clocks, like we wouldn't be where we are today if we didn't start out with that basic Horvath 353 CPG first generation clock, right?
Yeah.
And that's still very useful for certain circumstances, depending on the outcome, right?
Yeah.
So I think the limitation with the research in this field is when you're only looking at, one clock, right? We need to really understand what aging is and, the causation rather than correlation. I think we need to look at all of the clocks across the board. So that's why yours is even more unique, because you have this, systems aging clock, but then you have these, like, I guess you could call them subcategories, 11 clocks stemming off.
I almost call them like different aging dimensions if you think about it, right? There's different aging dimensions of your body because you're not aging in one dimension. You're not sort of unidimensional, so to speak, of in your biology, and that's what's really interesting. And I guess that's what's been sort of previously been missing, right? Like all of these clocks are amazing, but they were capturing a handful of dimensions really well. And now, since they're all separated out in the systems age framework, they are kind of like right in your face, I guess.
Yeah, definitely. And one question I had when you were talking about how you created these clocks, what cohort of people did you use for your study, or what did that look like?
Yeah, so we had two particular training data sets that we used in two different steps of our training process. So we had the health and retirement study, which had about 10,000 plus individuals that we used for sort of categorizing our different biomarkers and then sort of building these predictors using the epigenetic data. And then in the Framingham Hot study, which had about 4,000 something individuals, or a little less than 4,000, we used it to sort of train these scores and the clock ages and eventually the whole systems age as well. So we used both of these for our training purposes. And then we did a lot of validation as well. So we went to sort of the Women's Health Initiative study, where we sort of showed a lot of our insights around, and again, Women's Health Initiative was kind of like a blind data set for us. That's where we really showed, the improvements that these clocks really provide and kind of the agiotypes information. And that had about 5,000 samples. And then we're also sort of looking at the SATSA data set, which is a more longitudinal data set to show that longitudinally how these things change.
Yeah, and I don't even know if I've spoken with anyone about this yet, but can you just give our audience a general explanation of why it's so important to use a different data set for, I should say, training data set, right, and then a validation data set? Do you just want to explain that a little bit?
Yeah, sure. I think this goes back to some of the very sort of basic principles of machine learning, that you can essentially overfit to a data set, and you can overpredict, and your results might be exceptional in that data set, but they might be completely irrelevant in another data set because your features were selected, or your essentially what your methylations in this case was selected to predict really well in this particular data, but are not really relevant for another data. So what you really want is the data. What you really want to show is that, hey, you're not performing just well in the data that you trained in, but you're actually performing exceptionally well in a data that you had not touched at all while in your training. And that's kind of the standard in machine learning, so to speak, which is why kind of we use two training data sets altogether to create kind of this diversity, so to speak, of in our data, while also keeping like a completely two different blind data sets that we have not touched at all during our training data. training exercise, because you don't want to sort of bias yourself in any way.
Great. Thanks for that explanation. Just wanted to point that out in case, people had questions about, why would you use a different data set for training and validation? I'm going to go back a little bit and ask a question regarding maybe this is before you decided on the 11 kind of dimensions of aging, like you mentioned, but did you have a hard time or any issues with some particular biological system scores? I think, some metrics may have been imposed in this cohort, like better brain age prediction may have led to lower heart rate prediction, or, those strings were pulling in opposite directions in ways you didn't think they would act. And then how do you account for those changes or those issues?
Yeah, so I think that's partly like in some ways the limitation of what we work with
Okay.
is the data that we have, right? And so within the limitation of the data that we have, we'd like to assume, again, the health and retirement study is supposed to be sort of a cohort that is very representative of the US population. So we would like to believe that we were capturing all potential interactions that could have been there. But that being said, that is not going to be, it's only representative. It is not exactly the same human sort of US population. So I, for one, cannot say this with certainty that, hey, we've captured all dimensions, that, this is the best representation or the best weights that are there. That being said, what I can say with a lot of certainty is that given the data that we had and given the training models that we built in our sort of more blind data sets, we were able to sort of create the same validations and get the same really high accuracy. Of course, as you pointed out, there could be issues around, hey, in this particular data set, that maybe there's not enough people who have brain aging, or this particular data set, maybe a lot of people have cardiovascular aging. Those are all very reasonable, I think, questions. And I think the best way to sort of solve them is sort of keep improving these clocks with even bigger data set cohorts. So in some ways, right, that the more data that we have, the more people try these epigenetic clocks, the better we will be able to build them, the more information we'll be able to capture. And so it can only sort of improve from now on. And then in a lot of ways, right, for us, systems age is kind of the beginning of a framework. It's a framework for people to start really like going crazy on this, right? Like we want people to, and in one of the reasons why it's taken us a while to sort of get this out there is because we really want to standardize the pipeline so that anyone can just go and train a clock in whichever dimension of aging that they want to, and sort of build atop that as much as they want.
So I'm going to pick out a piece there. That was an amazing answer. But you said, you just said at the end there, normalizing kind of the effects, or I just lost my train of thought there. But the normalization pathways and using these algorithms, that way people can use them in different studies. Can you explain what that is and what that means? Because, something within my company at True Diagnostic is what we've worked on time and time and time again, is normalizing results due to batch effects or due to different issues, right? There's a lot of computational biology and science behind that. So can you talk about that? That's very new to our listeners.
No, I think standardization is kind of the name of the game here, right? Because what you really want is your again, going back to the whole point of using different data sets, going across these different studies, is primarily because what you want to build is something that works across different sort of populations, different groups of different ethnicities, of different, socioeconomic backgrounds, so on and so forth. And so unless you're really sort of standardizing the process through which you're doing sort of all of these algorithms and building these algorithms, what might end up happening is you're just kind of overfitting to a data set, and you might get like exceptionally good results, but those results are all farce because they have no value in the real world. And at the end of the day, all the science that we do is to benefit patients, is to benefit people. And if it's not really, it's all bogus if it's not really benefiting them. And so what you really want to do is kind of work or build these algorithms such that that they are very explainable, that people can, someone else wants to understand this algorithm and can build atop it. B, they are very reproducible, right? That someone else can really sort of just take the whole thing and retrain it and get the same results again and again, right? And C, what you really want is it should be something that it can be built upon, right? So now that we have spent these years building this particular thing, I don't want anyone else to spend another 3 years just rebuilding this, right? You just take what I've done and go crazy with it, build something even better. I would love that. And that's kind of the thing, right? You want to standardize the process of these algorithms that you're building, so that at the end of the day, all of us want to do good science and sort of build better for the world, and it can be as collaborative as possible if there is a standardization there.
Yes, that was the word I was looking for, standardization, when I my train of thought. That was broken down perfectly. Points A, B, and C, you're really spending so much time on making these usable, right, for everyone. So I can't wait to get my hands on them and see them when you're done. We appreciate all the work that you've been doing for this field. So now I want to talk about the applications of this clock and get into that a little bit. I think these clocks would be very interesting in interventional studies, obviously, right? Kind of like we hinted at the beginning. And you said, these one of the applications of these system clocks and why they're so important is because, people with similar epigenetic ages for first generation, second generation, or the gene didn't pace could actually have different systems aging. So yeah, what could people use this for? Like, what do you see it being most used for in the future?
Yeah, so I think I see this at sort of multiple different levels. I think at the very sort of trivial and basic level is where it could be something that is very consumer focused, where individuals go to a doctor and say that, hey, I want to know what my biological age is and what different organ systems are aging for me. And the doctor gives them sort of this single blood jab that gives them a vial of blood and tells them that, hey, these are the different organ scores that you have. These are the different rates at which you are aging. And hopefully now you can practice these behavioral or these, drug-based interventions that can slow it down, that particular aging. So that's kind of like the most simplest version of how this can work. The version above that, which I feel is kind of in drug discovery and in sort of building better interventions, right? So, think about it this way, that let's take an example of metformin, for example. We know that metformin, at least in retrospective studies, have shown to decrease cardiovascular disease events, right? But do we know for a fact, would that necessarily decrease the whole body aging at the same rate? We don't know. But maybe it just focuses, its effects are primarily on heart aging. So if you run a clinical trial that is capturing a whole body aging, you might not see any significant difference, and your whole sort of billion-dollar clinical trial goes down the drain. But in reality, maybe there is actually a very specific heart aging that is happening, or a decrease in heart aging that's happening, that you really want to capture. And there is a significant difference. And so what we can really do is kind of build more targeted interventions instead of like these more general interventions. And again, I'm not saying that eventually there will be, I feel like there will be interventions that are more whole body, but I think the place where we are at the science of aging, I think we will start more with very targeted organ specific interventions, like decreasing either musculoskeletal aging or decreasing brain aging or decreasing heart aging, so on and so forth. And that's where kind of these clocks become really, really useful. And then I think a level above that is when, so till now, we are looking at these clock scores kind of independently of each other. Things get really interesting when you start looking at them in tangent to each other, right? In congruence of each other. And that's when things start getting really interesting, in my opinion. Things start getting predictive, in the sense that now you can start predicting that, hey, you're in this sort of aging subtype, you're really predisposed to these XYZ diseases, right? And so you really need to focus, like, this is your risk score of developing this X disease in the next 10 years, or this is your score for developing this Y disease in the next 10 years. So let's really try and prevent that, right? And then the one level above this, and that this is where, again, things go a little crazy, and this is more where hopefully at some point we'll be, is you can not only you're just now looking at data very cross-sectionally. You're saying that, hey, this is what your aging score is right now. But with some of the longitudinal data that we have, what we're really trying to show that, hey, this is your aging score right now, and these are your aging scores, how they're going to be in the future if you don't practice any intervention. And these are your aging scores going to be if you practice these interventions. So actually predict that, hey, if you do these things, right now you're plus 2 years of age in your heart score. If you practice these sort of, say, behavioral interventions, you would be at -2 in 2 years from now. And that's where, things are really, really predictive. You're really able to sort of go into the nitty-gritties of that preventative medicine paradigm. And I hope that we get there very soon.
Me too, for sure. That would open up so many different doors and windows, again, for preventative healthcare and different screening. So I think it's, this DNA methylation that we're looking at in this epigenetic field, it just becomes more and more enticing and interesting if we can prevent the onset of disease and push that back, increasing health span, of course, because I don't want to live forever if I'm disease driven. So that's really the goal. And it seems like the sky's the limit. There's a ton of applications for that systems aging clock. So I wanted to ask again, are there any other reasons why people should look at the systems aging clock over traditional epigenetic age clocks? Anything you want to add there? I know you mentioned, of course, you may be you may be aging at different levels according to that system, but anything else?
Yeah, I think one other point was kind of just the balanced score, right? Which is some of these clocks are really trained well to predict certain dimensions of aging, and systems age, at least from the attempt that we have made, is able to sort of create a more balanced score that is not really biased in one particular dimension. So, what I'm really saying by that is, say, there's clock A that's really good at predicting cardiovascular aging, and say lung aging or something else. And clock B is really good at predicting some other type of aging. What's really happening then, it's just kind of over predicting for those agings in those dimensions, and you're kind of getting the not the most appropriate whole body aging. They're kind of like overly fit to that one dimension of whole body aging. And that's not what you really want. You want your whole body aging score, not the aging score of a few dimensions. And so that's the place where systems age can be really beneficial. And again, the other thing is kind of this identification of agiotypes, right? I think being able to categorize yourself into agiotypes and knowing that, hey, my agiotype is predisposed to these XYZ diseases. And so those are the ones that I really need to be on the lookout for. It's, again, super insightful. And the other thing that I would say is, and that's where it kind of, just because we have data that allows us to do this, we have done validations with that have shown that from about for 8 years of follow-up, these clocks, the scores are very predictive of sort of organ-specific disease events. So, for example, the cardiovascular clock is very predictive of cardiovascular disease, of myocardial infarction, so on and so forth. The lung clock is very predictive of lung cancer, the hormone clock of breast cancer, so on and so forth. So what we're really saying then is that these clocks, given the fact that they are more new and have been validated on larger data sets, are actually potentially, better predictive as well of future disease occurrences. And I think those are all sort of the pros, so to speak, of the systems age framework.
Yeah, and the systems age framework really takes the guessing away, right? Right now you receive this biological age score back, but a lot of times you're hypothesizing why you may have accelerated aging or deaccelerated aging, and you may be looking at epidemiological correlations and things that have been shown in the research. But I think the systems approach that you have is very unique in the fact that you say, aha, here's what's aging. Here's what I need to do. I understand my risk. What am I going to do about it, right? Or what is there to do about it?
No, exactly. So I think like one of the ways I would initially sort of explain this to people is like, hey, you went for a biological age test, whatever kind, not just epigenetic, and they gave you a whole body aging score. So what? What do I do with this?
Exactly.
Well, how do I intervene, right? But now oh, my heart is aging, so maybe I need to practice behavioral interventions that improve my cardiovascular disease, decrease my cardiovascular disease risk, or so on and so forth, right? So now you have a more targeted understanding of how to really intervene.
Yeah, the systems age is truly an overarching biological age, like this pure aging signal that is fundamental and encompassing of all of these processes.
Yeah.
So super exciting work. Well, Raghav, we're almost getting to the end here. What's most of your time taken up by right now? Is it really pushing this paper, finishing this up, I'm assuming?
Yeah, no, I think we're in the final stages of wrapping this paper up. There's some really cool additional analysis that I really want to do that I'm working on right now, just kind of comparing how some of, one of the things that's been out there about epigenetic clocks is that people say that, hey, why do we really need epigenetic clocks when we already have these biomarkers, very well validated biomarkers like, say, for cholesterol, for heart aging. So one of the things that I want to show is that, hey, we're actually capturing more information than just a simple cholesterol test or a simple, a simple heart repair rate test or something like that, or some very common scores like the ASCVD score, which is very common in sort of cardiovascular aging. And so if I'm able to show that, we can really very standardly say that, hey, epigenetic clocks built in this manner are actually more meaningful and insightful than the present biomarkers out there. And that, in my opinion, would really change the paradigm that we have right now, which people keep on questioning that, hey, okay, you have an epigenetic clock, it's not as good as everything else that's out there. But hopefully, if you're able to show that, that would be, I think, really a paradigm change.
Yes, I love that. And I need that data. Yeah, it's hard to convince, people who have questions about that. And it's great. They're asking the right questions, right? We need to prove that and show that data. I've heard even some experts say that the DNA methylation and epigenetic testing is going to overtake your conventional blood testing or your hormone panel testing one day, right? Because of the insights it can hopefully show and what you're working on. And not only the insights, but identifying those insights or issues earlier. Again, all coming back to the therapeutic preventative approach too.
Yeah, and I also feel like just the simplicity of it, honestly. Instead of being jabbed like 10 different times for like a metabolic panel and a renal panel and and another brain panel or a hepatic panel, now you're just being jabbed once and you're like, hey, I'm done. I'm good for now. And this, I know this sounds very Theranos-y, but I promise this is not like Theranos.
People ask us that all the time. Actually, we got asked that one time when I was on a call and I was wearing my hair up with a black turtleneck. And I was like, oh my gosh, I look like Elizabeth right now. It's like, I promise. It was some weird coincidence and I just had to say it.
Backing this up. Promise. There's valid science backing this up.
Yeah. No, that's hilarious. But yeah, I get super excited about the work. And you mentioned, only one stab instead of all of these different labs. And that becomes super, frustrating. And then there's a lot of education behind all of those labs. And then also the cost.
Exactly.
Like, the cost is going to go way down. I have it written on my website. Like, if you don't understand epigenetics, don't worry. We didn't understand genetics that long ago, right? And now we have the whole human genome sequenced, you can, pay less than $100 to get your entire genome sequenced in less than like 24 hours.
Yeah.
So if you think it's expensive now, the pricing is going to really decrease as work like yours becomes available.
Yeah, no, I think that's the way forward as we discover sort of new technologies that can process this epigenetic data faster and better and more accurately. The cost is only going to go down. I don't see a future where maybe say 5-10 years from now, it's just $10 worth of an analysis, and compared to sort of the canonical clinical biomarkers that you do, those panels are like hundreds to hundreds of dollars at this point.
Definitely. We'll hope to see that day.
Fingers crossed.
Yeah, fingers crossed. My last question, this is one I ask at the end of every single podcast. If you could be any animal in the world, Raghav, what would you be and why?
If I could be any animal in the world, that's actually a very interesting question. I think I'd like to be a turtle because they live the longest, at least like live fairly long, 200 years or something. I'd like to see 200 years of the world changing.
Yeah, no one has said a turtle yet, so that's like massive turtles at the zoo, and here's someone, a zookeeper speaking. They've been on this earth for like 150 years, and you're like, what? And they move so slow, and they're massive.
Yeah, and so I want to see the world change for 200 years, so that would be really cool.
Yeah, and all your work changing the world, right?
Fingers crossed.
Well, perfect. We've come to the end of this amazing podcast interview. I can't thank you enough. And for any listeners who want to connect with you, where can they find you? I'll put everything in the show notes too, but where could they connect with you or learn more about you?
I'm on LinkedIn, I'm on Twitter. And of course you can reach me at my email ID. That's my first name second name at yale.edu. And well, if you're ever in New Haven, just drop me a message. I'll be more than happy to sort of take you around and talk more about epigenetic clocks.
Awesome. Well, yeah, thanks again, Raghav. And thank you, everyone, for joining us at the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more. Thanks again.
Thank you, Hannah. Bye-bye.